Real-time PCR is a targeted method that measures a few selected genes. But what if we want to look at the whole transcriptome? Well, that's where RNA-seq comes in. A technique that allows us to profile the entire transcriptome and see which transcripts are present and how abundant they are. RNA-seq is a high-throughput sequencing technique that provides a genome-wide view of gene expression. Instead of focusing on a few selected genes, it allows scientists to examine thousands of transcripts simultaneously and compare their expression across different tissues, developmental stages, or biological conditions. Methods such as qPCR are highly precise, but they are designed to measure selected genes. Microarrays can examine many genes simultaneously, but they depend on a predefined probes. RNA-seq takes a different approach. It sequences the RNA molecules present in the sample, allowing researchers to profile the transcriptome without starting with a fixed list of genes. Taking plants as an example. The process starts with a plant sample such as leaf, root, or seed. Scientists extract RNA from the tissue, which represent the transcripts present at the time of the sampling. The RNA is then converted into complementary DNA or cDNA and prepared as a sequencing library. The library is loaded onto a sequencing machine, which generates millions of short sequence fragments called reads. These reads are then analyzed using bioinformatics. They can be aligned to a reference genome or transcriptome to determine which genes or transcripts they originated from. The number of reads associated with each gene is then used to estimate transcript abundance and gene expression. Genes with higher transcript abundance generally produce more reads and vice versa. By comparing samples, scientists can identify genes whose expression changes between conditions. For example, roots versus leaves, mutants versus wild type, young versus mature tissues, or treated versus untreated plants. These changes can be then analyzed to identify biological processes and pathways associated with the condition being studied. Plant traits and responses usually involve networks of genes rather than individual genes. RNA-seq allows researchers to examine these changes across the transcriptome and identify candidate genes, pathways, and regulatory responses. In plant biology, it's widely used to study development, tissue-specific expression, defense responses, mutant phenotypes, and responses to environmental or experimental conditions. It can also help generate candidate genes for further functional studies and breeding research. So, RNA-seq provides a transcriptome-wide view of gene expression. The workflow starts with RNA extraction, followed by cDNA and library preparation, sequencing, and bioinformatics analysis. The result is a profile showing which transcripts are present, how abundant they are, and how their expression changes between samples. That is the basic principle behind RNA-seq in plant research.